Our guest speaker today for this ICTR Enrichment Seminar is Professor Paul Shapiro from the UNB School of Pharmacy, Department of Pharmaceutical Sciences. Dr. Shapiro is the School of Pharmacy's Associate Dean for Research, and he also serves as the Associate Director for the ICTR Drug Discovery and Development Corps and the co-director of our ICTR ATIP Pilot Grant Program. Also, Dr. Shapiro was recently named as an Entrepreneur of the Year for the campus. Congratulations, Dr. Shapiro. Maybe you can provide some highlights about that award throughout your presentation. So please welcome Dr. Shapiro and presenting the NHLBI Catalyze program, providing bridge from basic to clinical researcher. Thank you, Dr. Shapiro.
Thank you, Wanda, and good afternoon, everyone. And it's a pleasure to present at the ICTR seminar series. So what I want to do is talk a little bit about a grant program through the Heart, Lung, and Blood Institute called the Catalyze program. I'm going to tell you a little story about the project that we recently received funding for, and how it's really provides resources to move products closer into clinical applications. And I also want to emphasize the support that I received through the ICTR to help facilitate that. So the outline of my talk is here. I'm going to talk a little bit about how the development and drug discovery of modulators, I call them modulators as opposed to inhibitors of kinase functions, these enzymes that catalyze the transfer of phosphate from ATP onto substrates and their role in disease. I'll specifically talk about a class of kinases called the ERKs, or the extracellular regulated kinases, and a target of them, a transcription factor complex called AP1, and its role in regulating airway smooth muscle cells in asthma. And then I'll talk a little bit about the support we received and how that helped generate preliminary data in a mouse model of asthma. And then I'll finally end up talking about this Catalyze program funding opportunity, which is called an R61, R33 mechanism. I'll talk more about that. And really its goal is to optimize lead compounds and get them ready for phase 1 clinical trials. So just as a little background on protein kinases and disease, in many cases, these enzymes are dysregulated through either activating mutations or through constitutive activation through extracellular signals, and their roles play this constitutive activation is involved in proliferative disorders like cancer, inflammatory disorders like asthma or other types of inflammatory disorders, and a whole host of disorders, neurodegenerative, developmental, and other that these kinases play important functions for, especially when they're dysregulated. And so we focus on this class of enzymes called mitogen-activated protein kinases. And today I'm going to focus more on the extracellular signal-regulated kinases as a subfamily of these. The Entrepreneur of the Year award that Jeff Hasday and I were fortunate enough to receive involved another class of mitogen-activated protein kinases called the P38. I'm not going to talk about those today, but the concepts that we use to develop compounds to modulate that activity of those enzymes are similar to what I'll tell you about today. But the bottom line is that they respond to extracellular signals to cause a cellular response. And it's dysregulation of these kinases that we're trying to modulate and try to affect. So just as a little background on the signaling mechanism. So from the membrane receptor, there's a signaling cascade through phosphorylation events, extracellular signals that regulate the ERK proteins, and they really have diverse functions, and they can modulate and regulate more than 200 substrates. And those substrates can do a number of things. They are involved in proliferation, inflammation, a whole host of cellular functions, including negative feedback. And again, this is a way to maintain the steady state or the homeostasis of the system. And it's dysregulation of this system that is involved in clinical disorders. Now, there's been a lot of interest in developing kinase inhibitors, and a lot of efforts have been generated to develop kinase inhibitors, and an obvious way to do that is to prevent them from catalyzing the transfer of phosphate from ATP onto a substrate. And so most kinases inhibitors that are in the clinic target the ATP binding site or the catalytic site, and they block all of these functions, including negative feedback. That comes with some consequences, and invariably, patients will develop resistance to these kinase inhibitors. The ideas that we thought would be maybe more beneficial would be, you don't need to block all of the functions of these enzymes, because some of these functions you might want to retain. So the whole idea is, can we modulate certain activities of these kinases so that we only affect substrates that are involved in clinical situations that we want to try to alleviate. So we call these, I like to call these more modulators as opposed to inhibitors, because they're modulating the function. They're not completely blocking the function. So how do we do that? Taking advantage of the structure of these kinases. Many kinases have a very similar three-dimensional structure, and that's what's shown in this ribbon structure here, where they have a domain up here that is where ATP binds. This is called the N-terminal domain. This yellow region here is where ATP or one of the residues that are involved in coordinating ATP binding. And as I mentioned before, most of the molecules, most of the drugs that are in the clinic, this is where they target. They block all functions. But kinases are very complex in their substrate recognition, and they have other sites that are involved in substrate binding, shown here in the green and the blue and the brown region. The activation loop, these residues shown in red here, these are what help activate these enzymes through phosphorylation of an upstream kinase. So again, it's a kinase cascade. And when they get phosphorylated, there's a conformational change that allows the transfer of phosphate from ATP onto those substrates. But we were more interested in these other regions that are involved in substrate interactions, and they have specific names. Some are referred to as the D domain. D domain is just referring to a specific sequence on the substrates that recognize these regions over here. So those are referred to as the D domain, or the recruitment sites, or the DRS. And then this other site here called the F site recruitment site, and they again recognize specific amino acid sequences on substrates that utilize this site for when they get phosphorylated by this kinase. So the bottom line is that these recruitment sites here regulate substrate interactions. So they asked the question is, could we utilize those in developing compounds that modulate the activity in a clinically beneficial way? And in particular, we were interested in this region called the F site recruitment site, or the FRS, because mutagenesis studies and previous studies by a number of individuals demonstrated that specific transcription factors, and particularly of the FOS family, were utilized this site when they were regulated by this kinase. And in particular, the FOS family is important because they are part of the activator protein 1, or the AP1 complex, that is involved in regulating a number of cellular functions. So we decided that this would be a place to attempt to develop compounds that modulate AP1 activity. And we utilized computer-aided drug design. My colleague, Dr. Alex McCrell here in the department, is an expert in helping design compounds that fit in these, that are predicted to bind to these various regions. And so we went ahead published some studies earlier. This is another study that published some compounds that targeted this site. And this is just a cartoon rendition of a compound fitting in that site. And basically what the computer-aided drug design individuals can do is they can look at how the molecule fits in here, and then make predictions on how to make that compound better. And so these these colored cages that you see here are predictions of different type of constituents that you can add to your molecule that would predict it to bind better and hopefully improve the potency. And so these are just an example of some of these compounds that are predicted to target these sites. And the computer-aided drug design can give you some numerical values to predict the potency or how much more they would bind. And this delta ligand grid-free energy score, so that the more negative the number, the more the computational predictions is that these compounds are going to bind better due to the modifications that are suggested. And so we can use these CAD predictions to make these compounds, and we give them these names. The SF stands for Steve Fletcher, who is a colleague and medicinal chemist colleague that helps synthesize these compounds. And so we can take these now, make these compounds, take them into the experimental models, and see do they indeed improve the potency of these compounds. So the red one here they indicate was a parent, we call a parent compound, and then these other ones are analogs of that. So we designate the ligand grid-free energy score to 0, and then everything that's negative to that is predicted to be more potent. And we can do some biological testing. Here we took some of these compounds again into biological assays. These are some cancer cells. These are melanoma cells. And just looking at viability, and you can see that they have these dose response curves, and the compound that was predicted to have the highest potency based on the CAD also was suggested to have higher potency in this viability assay. One compound here had no activity at all, and that's going to be important later in this talk, because I'm going to tell you a little bit about how this compound interacts with the target. And as you can see, this compound that has no activity has lost this double bond here, and whereas the bioactive compounds have this double bond. And it turns out that these compounds are forming a covalent linkage with a specific cysteine that I'll talk about in a little bit. We were interested in their effects on activator protein 1 or AP1 activity. And again, this is just a dose response curve, a luciferase assay that reads out the transcriptional activity of the AP1 off the AP1 promoter. This was done in another cancer cell line, A375 melanoma cell lines, which have constitutively active ERK activity. And as you see, there's nice dose response, and the compounds that have the lowest ligand grid-free energy scores seem to be the most potent as well. We were interested in AP1 because in the context of diseases like asthma, there seems to be an important role in AP1 in mediating some of the pathological responses in the lung when patients acquire remodeling of airway during asthma. So we went into a primary airway smooth muscle cell line. So these are airway smooth muscle cells that are isolated from patient biopsies, so they're primary cells, and you can stimulate them with a growth factor like PDGF, which is reflective of what happens in the clinical situation, and get an activation of an AP1 reporter. And then we can show that with our compound here, this compound SF3030, you can inhibit that AP1 activity. The VX11E is an ATP competitive ERK inhibitor, and it too, as would be expected, also inhibits AP1 activity in these airway smooth muscle cells. So based on that, we decided to pursue this project in asthma. And just as a little bit of background on asthma, which many of you already probably know, it affects probably more than 8% of the population of the United States, millions worldwide. It's one of the or the largest chronic childhood illness in the country, affecting maybe 1 in 10, maybe 1 in 13 children. There are management therapies to treat inflammatory responses and airway hyperreactivity with glucocorticoids, bronchodilators, leukotriene receptor agonists, and biologics are becoming a more important part of the therapeutic management of asthma, using monoclonal antibodies against IgE and various interleukins. However, none of them yet have shown to have any effective effects on airway remodeling. And that is one of the clinical problems when these airways become increases in smooth muscle proliferation, and then when they constrict, then airway obstruction is enhanced. And there's no effective inhibitors of the remodeling process to date to treat asthma. So we were going to go after the airway smooth muscles with these compounds. Just a little more clinical data. These are biopsies of lung tissue from normal and patients that have various degrees of asthma. So the normal would be on the left column, the mild asthma patients in the middle, and then severe asthma on the right side. And what this study did, which was published about 15 years ago, is it looked at the activated forms of these MAP kinases in these asthmatic and normal tissue. And they looked at the activity of the ERK MAP kinases, the P38 MAP kinases, and then these MAP kinases called the C-Jun N-terminal kinases, or the JNK kinases. And I think what you can see here is the green is the reflective of the amount of that activated MAP kinase. And so with the ERKs, there's a lot of activated MAP kinase in the epithelial tissue and in the smooth muscle tissue. And there's quantification of these fluorescence shown in the graphs here. P38 is more localized to the epithelial tissue and less so in the smooth muscle. And the JNK MAP kinases, if anything, the normal tissue probably has more staining in the smooth muscle compared to in the severe asthma. So this was just a little more justification for going after modulating ERK activity in lung tissue. And so based on that, we wanted to improve these compounds that we took into the cell models, so that we can take them into in vivo tissue, in vivo models, and we went to the ICTR and asked for some support to help optimize these what we call function selective ERK inhibitors or modulators. And so basically the request was to help guide the optimization using the CAD, the computer-aided drug design services. And one of the things that we noticed about these compounds, and I alluded to it earlier, is that when you lost a double bond on one of the ring structures, you lost the biological activity. And that double bond, it turns out, was forming a covalent adduct with a cysteine on ERK. So ERK here, shown on the left, is just a model of ERK. It has 7 cysteine molecules, and they're indicated here by the numbers. In the green region is this region where we're targeting, that's the F recruitment site that targets that the substrates that utilize that are like FOS, that are regulating the AP1, are interacting. And it turned out that we published that the compound targeted this cysteine at 254. And it's right adjacent to the site where the substrate interaction site, and in fact, it's even part of it. Now, the computational folks went ahead and they made predictions on the probability that a cysteine could be targeted, and they have Dr. McCrell has protocols to look at residues within a molecule to say, is it targetable? Some of these cysteines are buried, they're not accessible. And they look, and Alex's group looked at the probability that a cysteine could be modified. And it turns out that there were 3 of them that were. One here at 161 and 166, and to our satisfaction, the compound or the residue 254, where our compound was shown experimentally to interact. Now, it turns out these cysteines at 166 and 161 are in the ATP binding site. And others have actually generated ATP competitive inhibitors that target this cysteine. They made it all the way to the clinic, but they were not effective, and they showed some overt toxicity. And it could be, again, because you're blocking all functions of this enzyme. So we didn't go after these cysteines. We went after this one that modulated the substrate interactions. So that was very helpful to get that that support. It also led to an in vivo model or a publication that we had that showed that these compounds were effective in an in vivo model of asthma. So you can give mice asthma, and it's one of the models that you can utilize is this house dust mite model, where you expose mice for 3 weeks to this house dust mite, intranasally to this house dust mite, and their airways, they will basically develop the same pathologies as humans do in terms of airway remodeling, inflammatory responses. And so what is shown here is just is some lungs, histological sections of lung tissue that have been treated with the vehicle or PBS control. So these nice open airways are shown here in these circles here, nice open airways when you just treat with our compound. But with the house dust mite exposure, you get this thickening of airways and thick excess mucus secreting cells. The arrows are pointing to inflammatory cells. And so you can see there's a whole influx of inflammatory cells with the house diet. But if you pre-treat these animals right before you give them that house dust mite exposure, you can prevent the remodeling of those airways with this compound. So again, the compound was given intranasally as well to these animals. Since we were interested in airway smooth muscle, we also looked at a marker of airway smooth muscle, and there's a specific protein, alpha smooth muscle actin, that stains smooth muscle, airway smooth muscle cells. And so in the PBS treated, you get the brown color is actually the staining of the smooth muscle tissue, and you can see there's some brown with the controls. Same thing with given with the compound, and then you can see with the exposure to the house dust mite, you get the thickening of the airways. You get more production of airway smooth muscle cells, and then that can be mitigated with pre-treatment of the compound within this lower right-hand panel. And that can be quantified here, just measuring the area of smooth muscle actin in the vehicle control, the compound only, the house dust mite, which has the most exposure or the most accumulation of smooth muscle, and then that can be reduced when you pre-treat with the compound. We also looked at inflammatory cells in this model, and so this same type of treatments where you can take bronchiolar alveolar lavage fluid and look at inflammatory cells. And we looked at leukocytes, eosinophils, macrophages, neutrophils, B cells, T cells, CD4 positive, CD8 positive. And as you can see with all of them, the house dust mite, as would be expected, enhances the number of these inflammatory cells in the BAL. And in all cases, the compound, pre-treatment with the compound can reduce the number of influx of those inflammatory cells. So that led to an application to the NIH, to the National Heart, Lung, and Blood Institute, that was going to further optimize these function-selective ERK inhibitors for reducing AP1-mediated airway pathology in asthma. I have to give a lot of credit to my colleague, Deepak Deshpande, who used to be at the University of Maryland. He's now at Thomas Jefferson University. He is an expert in the animal models of asthma. And we applied to this program called the Catalyze program, which was really a program that is aimed at developing small molecules for therapeutic use or biologics. And so this is just a definition of the Catalyze program. I'm going to tell you a little bit about it, but if you want more information, you can go to these websites here. It's through a mechanism called an R61, R33 mechanism, and I'll talk a little bit about the years that are spent on each component. And again, if you want more information on this funding opportunity, this is the designation here. So basically, the Catalyze program is really to drive new discoveries and new development of treatments for heart, lung, blood, and sleep disorders. And it can be devices, it can be drugs, and it can be small molecules, biologics, new diagnostics. They're heavily into having an educational component in these programs. So they want to help train, new scientists that can address these type of issues. So this is from the Catalyze program goals. And this is kind of an outline of what they really help facilitate. And so the bottom line is they take a concept, which could be a new small molecule or some biologics, some idea of where it targets, and take that through defining the product, preclinical assessment, and getting it ready for clinical trials. And so it can also be devices or diagnostic tools. We're more in the space of the small molecules, and I would say that we're probably more in the preclinical development where we already have lead compounds, and now we're going to optimize those lead compounds, take them further through animal testing, efficacy, safety, and really try to get these molecules so that they're ready for the phase in human testing. So this is just basically the goal of the Catalyze program and what they hope to achieve with this source of funding. So I'm just going to go through our specific aims that we had. So the first two aims are the R61 component, and the first aim is to take advantage of these computer-aided drug design technologies to help optimize these compounds that target these substrate binding sites in both the inactive and primarily in the active form of ERK. So the structures are slightly different when it becomes active. So the first aim is primarily computational and medicinal chemistry. The second aim is to test those in cell and tissue models, and primarily looking at things like extracellular matrix that are part of the remodeling process, proliferation, the AP1 signaling in airway smooth muscle cells, and also in lung tissue. And then the third aim is the R33 component. So each aim, and I'll talk a little bit more about how the evaluation process with the NIH occurs. The third aim is the preclinical testing and looking at these compounds in vivo and doing further toxicology, pharmacokinetics evaluation. The R33 aim 3 component is also encouraged to have what is called an accelerator partner, and that partner in our case is this company called Genie Life Sciences, and they are the partner that has licensed these compounds, are ERK inhibitors, but also the P38 inhibitors that Jeff Hasday and I are working on in other models of lung inflammation and lung injury. So, one of the aspects of this program is the development of milestones, and so I'm going to tell you a little bit about how there's a little bit more paperwork with this grant mechanism, but I think it's a good thing because I think it holds you accountable for reaching certain milestones and achieving certain goals. So we had to develop milestones for the whole grant, and that's what's shown here. So the first aim has certain milestones, again, mostly designing the compounds, synthesizing the compounds, testing their binding. The second aim, more cell-based in vitro studies. And then the third aim, mostly the in vivo studies and doing the preclinical evaluations like pharmacokinetics, the physiochemical properties, metabolic activities, off-target effects, and toxicity in that third aim. And we have time periods that we have to achieve these. But we were required to even do more. We had to create individual success measures for each of those milestones for each year. And so here's an example for our year one milestones and measures of success. And again, these were developed with the program officers at the NIH. And we went back and forth, to make sure what we were proposing was achievable. And I think that's important because we often try to do too much. And I think they're really trying to keep us focused on the project so that we can, at the end of the day, have something that might be relevant for a clinical trial. And that's just shown here for the first aim. So we have 3 milestones and we have success measures for each of those milestones, that are based on the computational and the synthesizing, a reasonable number of compounds. The resources for these grants are not unlimited, so you have to be reasonable in what you can propose, and then testing of these compounds shown here in the third milestone, both just binding to the target, and are they better than what we have, and then also looking at not only the covalent compounds, but non-covalent interactions. So maybe this is still a little too ambitious, but this is what we agreed to with the NIH folks. So a little bit about the Catalyze program activities. As I mentioned, there's a little bit more that you have to do, but I think it does hold you accountable. We have quarterly meetings that we meet with the program officers and then also a research team that I'll tell you about in number 2. There is an annual meeting coming up where we meet with other researchers that are funded through this mechanism. I think there's like 15 or 20 other groups around the country that have this R61, R33 mechanism, and where we can talk about our projects, we can share information. We can the quarterly meetings are mostly to review the milestones, timeline, any challenges that we have. We also talk about intellectual property and partnerships and commercialization strategies. We have been set up with a research team. So the Researchers Triangle Park Institute, RTI International, has project managers that we meet with at these quarterly meetings, and they help keep us on track. And they can provide us with assistance, technical assistance. They have experts in toxicology, medicinal chemistry, manufacturing, pharmacology, regulatory science. So I think it's really good that they provide this so that we can bounce off ideas and, again, help keep the program on track. And then finally, they emphasize a commercialization and entrepreneurial aspect to this application. And each year we have to develop a product development plan. Where is this going to go and how do we see it going? One interesting thing I had to do was a 90-second pitch where I had to record myself telling an audience why this is important and how I'm going to solve the problem in 90 seconds, which is not that easy. And so these are just some of the aspects of the Catalyze program and some of the things that we get to do. So I think this is, I'm going to end there, and I just want to acknowledge the people who, this is really a team effort, and my long-term colleague Deepak at Thomas Jefferson, who is a co-PI on this R61, R33 grant. Couldn't do this without him or without my colleagues in the department, Alex with the computational, Steve with the medicinal chemistry. Maureen has been very helpful with proteomic analysis in these models, in these asthma models, and of course the people who do the work, all the folks in the lab, and then of course the funding through the ICTR. We also had some funding through the Center for Maryland Advanced Ventures Life Science Fund. And then the R61 is this HL, and we also had a previous grant from Allergy and Infectious Disease that help with some of the preliminary data. So I think that's all I have. I'd be happy to ask questions. I think I see some chat. Let's see. David Weber says, would you mind sharing your slides with the group, particularly with how you outline your Catalyze grant? That would be nice. I'd be happy to do that, David. Yep, I can. I don't know how we want to do this. I'm happy to, but anyone who wants to know more about this Heart, Lung, Blood Catalyze program grant, I'd be happy to share that information. Maybe Wanda, as far as sharing the slides, I can send them to you or whatever. Okay, I got Vana will, yeah. Okay. Any questions? I ended a little early. Go Cal, go Cal. So Steve Fisher and I, 100 years ago, were at the University of Vermont. That's where the catamounts come from. So the decision on applying for R01 and R61, well, the R61 is a 3-year grant. It does have a higher, it's not, if you just, a modular budget, it's a $350K a year budget. So unless you went for an R01, I think from my understanding, the competition for the R61 might be a little less than for an R01 application through a regular standing study section. But I don't know if that's absolutely true, but I think if you're interested in developing a product, if you have any entrepreneurial inklings, I would go for the R61. The other question is, why not an SBIR? Well, that's a possibility. The SBIRs, the first phase is pretty limited. I think you absolutely need a partner for the SBIR. For the Catalyze program, you don't necessarily need that accelerator partner, because the first 2 years of that grant are to help you find one. If you don't have one, we just happen to have one. I personally think it's a good idea to have an accelerator partner in mind who they actually have to be accountable for that 3rd year budget. They have to pay 25% of that 3rd year. So what we had was a commitment from the company that said we will support the 3rd year 25% of the $350K that is allotted for that 3rd year. So, the SBIRs, most of it goes to the company. So if you have your own company and you can do an SBIR, that might be an option. I hope that addresses that, Svetlana. Let's see, Alistair, do you know of an analogous program from my other institutes and other? I was looking up that there are at least, I think, 5 sources of the Catalyze program. I thought they were all through the NHLBI, but I don't want to. I was looking for that exact same. I was curious about that myself. I don't know if I can't say for sure whether other institutes offer this mechanism. Let's see, Christy, your requirements for NIH making our science, but do you think like to an RFA for beings, do you think is, Christy, I'm trying to think if I understand what you're asking. Do you think that R61 is like a R01 to a RFA for beings? So I think I understand what you're saying. I think it's similar to that. That it is really a focused mechanism for drug development. So I think I would agree with that. And Bob just said that the NIAMS has an R61, R33 mechanism too. Thanks, Bob.
If anyone would like to be unmuted to ask a question, we can do that at this time as well. Dr. Shapiro, thank you so much for that presentation. And I really think this is a wonderful example of the services that the ICTR has, that we provide to researchers across the campus. We know that we have researchers that request ICTR support for Dr. Mackerel's core, Steve Fletcher's core, and Maureen Kane's core. And I know that there's cross-campus collaborations between the School of Pharmacy, School of Medicine, School of Dentistry. So it's been really, a wonderful experience connecting researchers together. Dr. Davis, did you want to add any comments?
Yes, so thank you, Wanda, and thank you, Paul. That was a wonderful seminar and presentation. I had one question. So when your compound proves to be successful in the preclinical and animal models, what's the pathway to getting into first in human studies? Did you have to address that in the grant?
No, because the Catalyze program kind of stops there. The assumption, I think, is that the accelerator partner will take over that responsibility. And they did that. So we had evidence that our accelerator partner had already done that with the P38 inhibitors that Jeff Hasday and I had developed. And they have provided the resources to take them through a phase 1 and are now in a phase 2. So I think the assumption is that's when the company, the partner takes over and provides the support for that.
Thanks, Paul. The bashing and crashing, they're building the new hospital for advanced medicine just outside. So.
Oh, I can't hear. Yeah, I heard there is some background noise, but I don't hear it. So yeah.
You must have a great filter.
Yeah. There was
Dr. Shapiro, there was another comment in the chat.
Yeah. So Zhao Feng said, was the R61 reviewed for our standing study section? To me, it was not. It was a panel that was an ad hoc panel. So and I don't think it was competed later among the R61s. I think each funding opportunity announcement had its application. So you were competing within just the ones that applied for that. So it was not, my understanding, it was not a regular study section panel. It was put together specifically to review these applications. All right.
Any other questions? So I just want to remind everyone that we do have a survey. The link to that survey is found in the chat feature. And we would appreciate any comments that you might have about additional topics that you would like us to bring to you. And with that, Dr. Davis, Dr. Shapiro, do you have any other comments that you would like to add before we close the session and give everyone the last few minutes to have lunch.
I'm just seeing that other institutes have this mechanism. Other people are posting, like the NCI, I think, has one. So I'd say look into it. It's yeah, I think it's an interesting mechanism. You do have a little more paperwork, a little more accountability, I should say, compared to an R01. At least I think so. But it looks like there's other opportunities for this mechanism. Yeah.
Wonderful.
And thank you to the ICTR, to ICTR. I do appreciate their role they play on campus.
And the link to ICTR resources for drug discovery development is also in the chat feature. So if you're interested, visiting with any of the cores, Dr. Mackerel, Dr. Fletcher, Dr. King, and Dr. Vallapalli, we can send you that way. Govaru, I'm so sorry.
It's okay.
Govaru.
Yeah. Right. Thanks, everyone.
Cool. Thanks, Wanda.
Take
Bye,
care.
everyone.
Bye.
Bye.